Okay, this is what we will do for this afternoon session. We'll talk about RNA tertiary structure. Okay, in our previous lecture, just to give a small recap, we all came to a common conclusion that there are a lot of factors that affects the formation of secondary structure. We have discussed in brief what are those factors and how do they affect the formation of RNA secondary structure. And then we concluded that the structure that is reduced in vitro condition, or the structure that might be predicted using several structure prediction program for RNA, well, they might not reflect the very reality that might be over there in cellular conditions as such, or in vivo conditions. Okay, so these things are to be kept in mind before you claim anything in your research. Okay, that was the main objective of discussing all those things. Okay, so now we have completed the primary structure. We have discussed the primary structure. We have discussed the secondary structure, the factors that affect the formation of secondary structure. Now it is time for us to move to another level, to the next level of RNA folding, and that next level is RNA tertiary structure. Okay, so well, how would you define RNA tertiary structure? Okay, just like protein, we can also define RNA tertiary structure as the specific arrangement of RNA secondary structures. Well, that might include helices and other structural unit in three-dimensional space. We have defined protein tertiary structure in the similar way. Okay, that is the specific arrangement of secondary structure that are involved in protein in three-dimensional space. We call it as its tertiary structure. Same is the case with your RNA. Okay, so the figure that you see over here on the left hand side is the tRNA secondary structure, or what we call as the RNA roadkill diagram. Okay, so why we call it as roadkill diagram? Because this is how the RNA would appear if you place it under under in any vehicle on road. Okay, so it chapta ho jata hai. So it is similar to something that that appears, the dead animal that appears on road, वो ऐसे चपटे हो जाते हैं road के साथ में. So that is why it is called as the tRNA secondary structure, or the tRNA roadkill diagram. And you know, most of the molecular biology book shows this tRNA roadkill diagram. Okay, so you people might be already aware of the tRNA's secondary structure. It has got 1, 2, 3, 4, and 5 arms as such. Okay, so this arm is called as D loop arm. Then you have got this anticodon loop. Okay, you have got this variable loop. You have got this phi U loop, and you have got this acceptor arm. Okay, so of course this anticodon arm is going to be the one that will go and interact with mRNA molecule, and over here you would be having your amino acid attached, right? So it is said that this is not the original state of the tRNA when it is present in cell. It is said that it would fold itself upon each other. The secondary structures that are shown over here, they would fold up upon each other in a specific way and give rise to a tertiary structure, which might be represented something like this, which is shown on the right hand side of the your figure diagram. Okay, so now the secondary structural unit usually undergo the process of folding to attain globular and functional state. As in case of protein, यहाँ पर भी ऐसा ही होता है. Okay, so unless and until protein is folded properly, it cannot attain its functional state. In a similar way, in order to perform its function, the RNA molecule is also supposed to get folded in a precise manner. Okay, so it is the process of folding that your RNA structure will get some globular state, and it will eventually help it to attain its functional state. Okay, now the major player that comes into picture over here is in form of a divalent cation ion, which are usually Mg dipositive ion. These divalent Mg dipositive ions plays a very important role in RNA folding. Okay, so just to make things clear, in primary structure, it interacts with monovalent cations like K plus ion that results in formation of secondary structural states. Okay, and after the formation of secondary structure, divalent cation like Mg dipositive comes into play, which eventually help it to obtain the globular fold and get some tertiary structure that can be functionally active. So it is K plus ion that maintains the secondary structure, and it is this Mg plus plus ion or Mg dipositive ion that would eventually result into formation of tertiary structure. Okay, so now one might ask why nature might have selected Mg dipositive ion in RNA folding. Okay, so there are a few reasons behind this. Okay, first one being this Mg dipositive ion can coordinate and tightly pack 6 chemical species in octahedral geometry. As you can see in this picture, you see over here a centrally placed Mg dipositive ion, which can coordinate 6 chemical species in its coordination state. Okay, and these 6 chemical species can be over, we'll say for example, uh, these are 3 water molecules as well as 3 phosphates, as you can see over here. Okay, so this Mg dipositive can increase local rigidity of RNA by tightly packing the functional group. Okay, so this Mg dipositive coordination would tightly pack oxygen atoms that would impose oxygen distance to around 2 angstrom. Okay, so since these oxygen atoms would be placed around, well, say for example, with a close distance of 2 angstrom, such short and strong coordinate bonds will impart stability to compact 3D structure of RNA that is usually not possible by monovalent cation like K plus ion. So that is why it is necessary that there must be Mg dipositive ion so that RNA can get its final folding and assembly. Okay, so this is where your Mg dipositive comes into picture. Okay, good. Now let us talk about,
Sir, can you explain this again?
the thing is ki dekho, if there are only monovalent cations like K positive ions which are present, so K positive ions, uh, are able to maintain some specific distance between the regions of your secondary structures. Okay, but in order to fold it more compact and fold it more globular way, okay, it is necessary that the secondary structures again come close in vicinity of each other, right? So this function is usually done by this Mg dipositive ion. Okay, why? Because first its coordination chemistry is in such a way that it can coordinate 6 chemical species around itself. Okay, and if these oxygen atoms, whether they are in form of your phosphate group, okay, or whether they are surrounding water molecule, so when Mg ions comes into picture, well, they can come in more close vicinity of each other, and that would result in more tightly folding and it can help it in obtaining more compact nature as such. Okay, so Mg dipositive ion would result in short and strong coordinate bonds, and that imparts stability to the compact 3D structure. And it is not the character with the K plus ion. Okay, therefore, Mg dipositive ion is essentially required for obtaining the tertiary structure for your RNA folding as such. Okay,
Okay, sir.
so let us, people have also placed a kind of mechanism by which this tRNA can fold, right? So it is said that initially coaxial stacking occur when your tRNA first encounter Mg dipositive ion. Okay, and how this happens, I will tell you. First, the short duplex stack on one another to form long and contiguous duplexes. Okay, well, say for example, your psi loop, which is shown over here in yellow color, well, this particular stem will stack over the acceptor arm. Okay, and simultaneously, your purple colored D loop will stack over the anticodon arm और एंटीकोडॉन लूप. ओके, तो ये सारे ये जो दो चीजें होती है, ये they happen simultaneously, it is said. Okay, and after the staking, which is mediated by this Mg dipositive ion, they eventually this Mg2 ions bring about the strong coordinate interaction in such a way that your RNA finally gains the final globular structure. So this is how your tRNA can obtain the specific folding that would enable it to transfer the amino acid from cytosol to the site of protein synthesis, right? Okay. So now there are some popular RNA tertiary structures that have been solved, and they are known to be functionally associated with, say, some catalytic function. Okay, so we would be discussing only 4 of them. Okay, so the very first category of tertiary structure that is usually found associated with RNA is these pseudoknots. Okay, so these pseudoknots are formed mainly by base pairing phenomenon. Okay, so these pseudoknots are forms when there is formation of these concatenated duplexes. Okay, so if such a thing happen, concatenated and one of the neighboring one of the neighboring strand would go and base pair with its surrounding duplex as such. Okay, therefore we say that they forms concatenated duplexes. Okay, so what happens is that the loop of one duplex, which we are talking about here, whatever the bases which are present over here, pairs with nearby sequences of the RNA. Okay, the most prominent example is in form of your biotin receptor, in which you can see the adjacent strand of your RNA sneaks into the major or minor groove of the folded strand as such. Okay, so this is a typical structure of your biotin receptor. Okay, and it is form of a tertiary structure called as pseudoknots. Okay, then what you have something called as tetra loop receptor motif. Okay, so this tetra loop receptor motif was discovered by Doudna group. Okay, and this tetra loop receptor motif is present in folding motif in group 1 self splicing intron. Okay, so this tetra loop receptor motif, it has got ability to catalyze their own excision from the precursor RNAs. Okay, so what happens is that there is a highly conserved tetra loop which might have such sequence over here. Okay, so this highly conserved tetra loop will go and dock into its receptor site. Okay, and such docking, well, receptor site is usually in form of like GAA tetra loop sequence. Okay, so now this GAA tetra loop and its corresponding receptor, well, they are having, say, conserved sequences in both of them. Okay, so this tetra loop receptor motif is present in P456 domain of this tetrahymena ribozyme. ये जो स्ट्रक्चर दिख रहा है आपको, this is P456 domain, this much domain, इसे अपन क्या कहते हैं, P456 subdomain, and this entire structure is your tetrahymena ribozyme. Okay, so over here you can see that over here is the tetra loop which goes and binds to its corresponding receptor, which is shown over here in green color. Okay, and this is all part of group 1 intron. Okay, so after this docking event happens, that helps in self excision of this subdomain that become functionally active later on. Okay, so you can refer this paper which describe this group 1 intron and its self splicing events in more details. Okay, then what we have is called as kissing loop. So this kissing loop is formed when sequences from adjacent stem forms Watson-Crick base pairing. Okay, so it is very crucial for efficient reverse transcription process of HIV. Okay, so it might play an important role in forming the tertiary or quaternary structure of that particular enzyme. Okay, now it is its tertiary structure by which you can now guess why it is called as kissing loops. Okay, then another common type of tertiary structure is present in form of these ribose zippers. Okay, so the thing is that now we know that this OH group can form two hydrogen bonds. So in this way, such a kind of zipping type of arrangement was first found in group 1 intron from Tetrahymena thermophila. Okay, so what you can see over here is that it forms a bifurcated hydrogen bonding pattern between the 2' OH group on the ribose sugar, okay, and 2' and the different atoms on the corresponding opposite strand of the ribose sugar on the other side. Okay, so these ribose zippers have been observed. They might act as some sticky or gluing in nature, which sustains the stability of the tertiary structure. Okay, तो इस तरीके का जो हाइड्रोजन बॉन्डिंग पैटर्न है, we call such tertiary structure as ribose zippers. It acts as a glue. It holds together the strands in RNA structure. Okay, and it is all possible due to formation of two hydrogen bonds via its hydroxyl group. Okay, so that was all about some famous tertiary structures of RNA. Well, now let us talk about representation of RNA structures. Well, what are the different methods and what are the different techniques which people have devised to represent the structure of your RNA? Okay, so first of all, why is it required to have so many, structure representation method? Okay, so first and foremost, our human mind is not capable of comprehending problems which have large amount of data in strictly numerical or character format. And that is why it becomes difficult for you to memorize and remember the amino acid sequences, right? So in order to increase this comprehension of RNA secondary structure problem, many type of visualization ways have been devised. Okay, so there are various alternative formats that are enable that would effectively enable for understanding the depiction of who is interacting with what, and that would eventually improve the readability of the so form depiction of RNA secondary or tertiary structure. Okay, so that is why we need so many different ways of representing your RNA structure. Okay, so here are some popular forms of representing RNA secondary structure. Okay, so the very first and simplest form, or first or very initial or pioneering work on representing RNA structure, people usually use this string representation, जो कि नीचे बताया गया है. Okay, then came the circle plot. Okay, उसके बाद कुछ दिनों बाद secondary structure graph device हुए. There are some papers that show the base pairing in form of mountain plots, while some papers do show the secondary structure formation in form of dot plots. Okay, well, now let us talk about one by one in more detail. Okay, first and foremost we have string representation. Okay, string representation यानी क्या? Just represent your RNA sequence in a form of a string. Okay, it is just like your FASTA format. Okay, what is the advantage of such string representation is that it is simplest form for representing RNA sequence, where one letter code depicts the RNA sequence. Okay, what are the disadvantage in such a case is that there is no secondary structure information. अगर आपने सिर्फ ऐसा string representation में आपका amino acid आपका RNA represent किया, you really don't know कि where are secondary structure and how is the base pairing happening in this RNA. Okay, so it is very difficult to comprehend and it is very difficult to visualize as well. Okay, then people along with string representation, a new methodology was devised, what we call it as bracketed representation. Okay, sometimes it is also referred to as bracket and dot notation. Okay, so what we have is the simple string representation of RNA on one line, and the adjacent to it, what we would do is that we would open or close the bracket and place a dot for representing the base pairing that would happen in your RNA. Okay, ये जो ओपन और क्लोज ब्रैकेट है, ये क्या बताएंगे कि who I mean what residue is getting paired with what residue. Okay, so we'll say, for example, if you consider this C is getting paired with this G. This G is getting paired with something else. Okay, but now the rules are quite simple. Okay, if a bond exists, or you can say if a pairing exists between nucleotides at position I and J, then an open bracket, okay, like this is used at position I, and its corresponding closing bracket like this would indicate to whom that particular base is getting paired up with. That would be your jth position. Okay, so now if no base pairing exists between any two bases, we give a dot over there. Okay, to nucleotide position that represent that it is not bonded, or it is not base paired with any other residue. Okay, so the rule is for every opening bracket there must be corresponding closing bracket to represent the pair. Okay, so what are pros and cons? Well, the advantages is that it is the simplest way to visualize the secondary structure of RNA. Second advantage is that it is machine friendly. Okay, what are its disadvantage? It is very difficult for human comprehension. Okay, it is machine friendly, but it is not necessarily human friendly. Okay, it is very difficult to comprehend. You can really get lost ki where is the corresponding closing bracket for this particular G or this particular bracket. Okay, whether it is this or this or this or this. Well, it becomes kind of fuzzy. Okay, so it is difficult for human to comprehend as well as it is difficult to visualize, but it is machine friendly. Okay, so these there are some pros and cons which are associated with each depiction technique. Okay, then came something called as link graph representation. So in this link graph representation, the nucleotide bases are drawn on a line at equidistance interval like this, okay, on x-axis. Okay, now the arcs are drawn that indicate the base pairing. Okay, so for example, this particular residue is getting paired up with this particular residue. Okay, so now it becomes easy to determine if there are pseudonorms. Okay, now if there are some, if the arcs get crossed, then we can, determine if there are pseudonorms or not. Okay, so this is how you represent your secondary structure in link graph representation. Okay, then you also have mountain plot. In mountain plot, what we do is that on x coordinate, okay, is the position of k of nucleotide, that is the distance of nucleotide, and its corresponding y coordinate would represent its base pairing as such. Okay, so well, just to make it or keep it simple, the loops in your RNA secondary structure would correspond to the plateaus, while these stems would correspond to the slopes as such. Okay, so the closer the two curves are, the closer these two curves are, the better defined is the secondary structure. Okay, in late 1980s, people started to visualize the RNA secondary structure as RNA dot plots. Okay, so these dot plots are well suited to represent structural ensembles. Okay, we will talk about ensemble in a while. Okay, so what we do over here is that the sequence is arranged in x and y axis. The same sequence is arranged in x and it's corresponding in y axis. Okay, so wherever there is base pair, that is in terms of I and j, so wherever there is base pair, a dot or a box in a row I and column j is placed in a rectangular grid. So first you would create a grid, and wherever there is, base pairing, you would place a dot or you would place a any rectangular box, or it can be a dot. Okay, so initially it is a dot, therefore we call it as a dot plot. Okay, so this rectangular grid correspond to the contact matrix. Okay, so now what happens is that if there is multiple secondary structure schemes, well, say for example, this particular triangle would correspond to something called as its contact matrix, because it would give you a complete idea about how many possible combinations might exist that might show the base pairing between the same sequence. Okay, so the dot plots are generally used to generate the 2D schematics. Okay, so this is how it works. Well, say for example, if this is the secondary structure, so over here, as it starts out with first, second, and third, okay, this is the third, you know, uh, third residue or third nucleotide which binds, or you can say which pairs up with the last nucleotide. Okay, so on y axis, this is the third. On x axis, the same sequence, this place would correspond to the last. So this dot correspond to the pairing of the third position and the 60th or 62nd position as such. Okay, and similarly, if you move ahead, this particular diagonal would correspond to this particular secondary structural unit. Okay, similarly, this particular diagonal would represent this secondary structural element, and this particular diagonal would consider, you know, this folding pattern. Okay, so in this way, the diagonal would represent the folding pattern. Okay, good. So now it is also possible that there might be different ways in which the same structure can be folded, and thus it can be represented in a single representation or a single plot. Well, the green color represent one format of folding. The your red color shade would represent some different form of folding. The black color would represent some different form of folding. Okay, so it is possible to represent all possible folding all possible folding conformations in a single plot. Okay, and that is what we call it as ensemble. So multiple conformational states, if they are expressed at a single time, or you can say at a in a single file, you call it as an ensemble. Okay, then you have some complicated RNA structure representation schemes, and these are called as tree and forest representations. Okay, so now these schemes are used to store secondary structures compactly in strings. Okay, so the example of tree and forest representation are sequence annotation tree representation. These tree and forest representation include full tree representation, and they also can be in form of ship arrow style tree representation. I will show you this one by one. Okay, what you see over here is the RNA structure representation in a sequence annotated tree representation. Okay, so if this is the folding pattern of your RNA sequence, you can represent this particular folding in sequence annotated tree representation, which is shown in its right hand side. Okay, so such a type of RNA structure representation was introduced by Hoshman back in 2003, and this particular representation was implemented in program called as RNA Forester. Okay, and the objective of implementing such a kind of representation was finding out the local similar regions in the RNA secondary structures. Okay, so can you correlate it? As you can see over here, in a tree, this is your stem. Okay, where A is one of the leaf. Okay, this A is over here. Okay, which is one of the leaf, which is attached to this stem, and this U is another leaf. Okay, so this would be a stem that would correspond to this. There is an unpaired C over here, जो आपको यहाँ से उसका एक branch अलग हुआ, which is attached to this C is attached to a different branch, right? Similarly, over here, your tree got bifurcated into two major branches. That is what you can visualize it over here, right? And you have such leaves in place of the nucleotides as such. Okay, so this is called as sequence annotated T tree representation. Okay. Next, in 1993, Fontana et al. First proposed full tree representation. Okay, so in which we don't consider letters, but we denote the base pairing in symbols. Okay, so each base pair correspond to a interior node. Okay, while each unpaired bases correspond to the leaf. Okay, as you can see, this A is present as a leaf over here. Okay, and base pairing is represented as an interior node. Okay, so usually a virtual root vertex is added just to make it look like a tree. Okay, because trees usually have root. So this is one of the way of representing your RNA structure. Okay, then you have something called as Shapiro tree representation, which is very similar to your previously described schemes. Okay, so over here this RNA, if this is the scheme for your RNA secondary structure, or it can be represented, the bifurcations are represented in form of nodes over here. Okay, so you can pause at this slide when you watch your video and try to relocate all these bifurcations, so that you can note how this tree can be formed from these secondary structures. Okay, it is quite simple. You can do it by yourself. Okay, so the final topic that we are going to discuss now is all about the energetics that is involved in RNA structures. Okay, how do we contribute the free energy of RNA folding? That would be the main crux of the remaining part of our lecture. Okay, so now first let us discuss what are the factors that affect the stability of RNA structure. Okay, first and foremost, it can be hydrogen bonding between the base pairs that affect the thermodynamics or energetics that is involved in formation of RNA structure. Okay, and this hydrogen bonding between base pairs was used as a character to define a model that would be helpful for calculating the energetics involved in RNA folding. Okay, so this nearest neighbor method use this hydrogen bonding between base pairs as a criteria for free energy calculation. Okay, so such free energy calculation based on bases that can potentially interact with one another. While on the other hand, Turner's method relied on considering how the neighboring base pairs are stacked upon each other, and that would eventually give rise to some terms that can be used for calculating the free energy. Okay, so all in all, the Turner's method used free energy calculation based on stacked base pairs as such. Okay, so well, here is how things would work. Is that let us first define how free energy is calculated first. Okay, so the relationship for calculating the free energy in terms of enthalpy and entropy can be given with something like this. That is, delta G is equal to delta H minus T delta S, where delta G stand for change in free energy. Delta H is the term that correspond to enthalpy, and delta S is the term that correspond to your entropy, while T is the temperature. Okay, so now if we apply this particular equation for calculating the free energy change that happens when RNA gets folded from unfolded state, RNA helix formation at physiological temperature is usually at 37 degrees centigrade. Okay, so now the same equation can be rewritten by as delta G, that is the free energy calculation at temperature 37, is equal to change in enthalpy minus 310.15, that is the temperature, or you can say physiological temperature, into change in entropy, that is delta S. So now, as per the nearest neighbor model that we have already talked about, the standard free energy of helix formation for oligonucleotide is calculated by using three terms. So in order to calculate the change in free energy by using nearest neighbor model, तो आपको तीन चीजें पता रहना जरूरी होता है. The first thing that you should know is something called as the initiation free energy. So this initiation free energy is change in helix initiation association with formation of the first base pair in a duplex. Okay, so what this means is that there should be some term that would take account of what are the two bases that would form base pairing at the first time in while in the process of folding. Okay, so that is called as the initiation free energy. Then next, what you should know is the propagation free energy. Your propagation free energy is nothing but sum of propagation of free energy of forming each and every subsequent base pairing. Okay, so we'll say, for example, if your RNA is in unfolded state, there would be at least mathematically there would be a first base pairing that would happen, right? And the free energy associated with the first base pairing that would happen while the process of folding is going on, we would call it as initiation free energy. And once the first base pairing has happened and subsequently the secondary structure starts to form by subsequent base pairing of the remaining nucleotides, that would be used in calculating the propagation free energy. And finally, you need something called as the symmetry correction. You need to add the symmetry correction only if the sequence is self-complementary. Okay, we will talk about this thing. I would give you an example so that things would become more clear. Okay, so what happened is that this Doug Turner figured out the enthalpic and entropic contribution for RNA helix initiation, propagation and symmetry correction. Okay, ये जो तीनों टर्म अपने को लगने वाले हैं to calculate the free energy associated with RNA folding, this Doug Turner kind of standardized the values that can be used for free energy calculation. Okay, so whatever the free energy that are associated with propagation, initiation and self-complementarity, well, he calculated this value on the basis of on the basis, just a minute, on the basis of experimental data of 45 complementary RNA duplexes. Okay, so now here is what he has done. Okay, so just now consider that if A and U are already base paired, okay, and after this base pairing there is another base pairing of another A getting paired with U, and thus this base pair will stack upon the subsequent base pair. That is, AU base pair will stack upon AU base pair. Okay, so if such is the case, the change in free energy that is associated with such stacking would be minus 0.9 kilocalorie per mole. Okay, and subsequently he figured out for each and every possible combination of such propagation sequence. Okay, so इसे अपन propagation sequence क्यों कहते हैं? Because it is way in the middle. This has been already formed and इसके बाद में subsequently अगला propagating base pairing होने वाला है. इसके बाद में दूसरा एक base pairing and stacking event होने वाला है. Okay, so these values are to be used for propagation sequence. Okay, for initiation, okay, initiation, that is the very first base pairing to happen, we would have to consider the value over here. Okay, बात समझ में आ रही है आपको? तो ये सारे वैल्यूज कंसिडर करना पड़ता है. नाउ इट इज एंटायरली पॉसिबल दैट देयर माइट बी डैंगलिंग एंड. तो डैंगलिंग एंड के लिए यू कैन यूज दिस थिंग. Okay, so well, what do this value correspond is that there is base pairing of a and u, and there is some dangling x. Okay, so now if the dangling x is a, then the free energy associated with it would be minus 0.8. Okay, ठीक है, बात समझ में आ रही? So this is for 3' dangling end and this is for similarly 5' dangling end. So how these numbers are used would make things more clear. Okay, so we'll say, for example, if you want to find out what is the free energy associated with folding of this particular secondary structure. Okay, so what you would need is the initiation. Okay, we'll say, for example, this is a 3' 5' dangling दिख रहा है यहाँ पर. तो 5' पहले, हाँ ठीक है, 3' से शुरू करते हैं. So this AU is a base, AU is a base pairing, and over here X की जगह पर क्या है? You have got A, that is this situation. So what would be the free energy associated with such a kind of stacking? It would be minus 0.8. Similarly, on 5' end, you would have like this. You have got CG, which is shown over here, and in place of X, you have got A. Okay, and table में उसका corresponding position क्या रहेगा? This value. So if this C gets gets base pair with G with a dangling of A at 5' end, then free energy associated with such stacking is minus 0.5. Okay, so this was for the dangling end. Okay, all you need to do now is to figure out the propagation sequence. Now you know CG is then stacked upon GC. CG is stacked upon GC. Its corresponding free energy change is minus 2.0, which we have written over here. And similarly, we would go on calculating the stacking interaction and its free energy value associated with. This is called as propagation. Okay, so as per the equation, need for calculating the free energy, उसमें 3 terms होते हैं, right? The initiation free energy, the propagation free energy, and the symmetry correction. Symmetry correction only if the sequence is self-complementary. Okay, so here we go now. इसका initiation free energy कितना है? 3.4. Do you see? Why? Because this is the value that is needed for initiation. Then you would have to add up for all the propagation free energy values. इन सबको आपको add करना पड़ेगा at 37. Okay, whatever the propagating sequences are, and you would have to add the symmetry correction, that is 0. By why? Because this particular sequence is non-self complementary. Okay, now let us talk about how do we decide whether the sequence is self complementary or non-self complementary. Okay, now if you see that the sequence folds right over here, okay, then you say that the sequence is self complementary. And if it happens that this is just part of say some helix, you don't know कि ये कितना दूर जाकर fold होकर वापस आया है. In such case, you would call it as non-self complementary. Okay, so in such case, we don't know where this sequence folded or how far that sequence went or how far this propagation went before it got folded, before it got some U-turn. right? So in such a case, we consider the symmetry correction as 0, okay, because it is non-self complementary. We add it over here, okay. So once you add everything, you might end up with the total free energy that is released when such secondary structures are formed. It is quite very simple, okay. So now एक चीज और बच जाती है कि what if there are loops in your sequence. You would have to add the you would have to add the scores for the loops as such, and that would completely depend on the size of the loop, okay. So well, say for example, if there is a bulge which involve only one residue, you would have to add plus 3.3. Okay, well, say for example, in this case, how many residues are involved in this loop? 1, 2, 3, 4, and 5. So we would have to check the loop size, which is 5. And what is it? It is a hairpin loop. So consider this. So if there is a hairpin loop of 5 exposed residues in your loop, then you would have to add plus 4.4 in the final score, right? Like over here, you see that there is a bulge of one residue. Therefore, you would have to add plus 3.3. Okay, so these things are to be added to the final score, right? So this is what will account for free energy increments for your loop. Okay. So this is, just a basic example of how Duck-Turner method can be used for free energy estimation of RNA folding. Okay. So later on in 2003 or 4, this method was, revised with lot of modification. Okay. And the present form of mfold web server is implemented on such approaches. Okay. So there are two web servers that can be used for RNA folding. Okay, what we call it as the mfold web server and Vienna RNA web service, which also enables you for structure prediction, folding kinetics, sequence design, and so on and so forth. Well, when you go in your second semester, I would teach you on what are the algorithms that are involved in RNA secondary structure prediction. Okay, over there we would take into more details of this thing. Okay, so this is it for now. Okay.